If you're working in medtech and are struggling to make your devices more sustainable, then there's a link in the description for you. Our guest today is Arne Jaksch. Arne is an expert for toxicology and one of two managing directors of his own company, Jaksch Life Science Consulting. He supports medical technology, pharmaceutical, and chemical companies in the biological and toxicological evaluation of their products. And here is our discussion. Hi, Arne, and welcome to the podcast.
Hi, Lucas, dear listeners. Thank you very much for inviting me. I'm very pleased to be able to talk to you today.
Yeah, so Arne, you're an expert for toxicology and toxicological risk assessment. Can you tell us when and why one does a toxicological risk assessment and for a medical device or for another device?
So risk-based planning of biological safety assessments and toxicological risk assessments is a regulatory requirement, actually. If we stay in Europe, it comes from the IVDR and the MDR regulations. And therefore, you need to have one. You can't avoid it. And so much to the why. And when you would start with the toxicological risk assessment is depending a little bit on in which phase of the life cycle your product is currently. That's kind of tricky. The best thing is, of course, if we come in early, already with the material selection or shortly after the materials have been selected. And then we can address already the first questions which are going to come or influence still R&D decisions, which cannot be influenced as much at a later point of time.
Okay. So on the podcast, we've talked about chemical analysis. We've talked about chemical analysis of gas paths. We've also talked about the biological risk assessment. Could you put that in the overall framework of the biological risk assessment, the toxicological risk assessment? Where does it come into play? What do you do first and what comes after that? And which is the niche of the toxicological risk assessment?
Mm-hmm. So if you're talking about the biological safety risk assessment, usually that contains a plan and then a verification phase in this case. And the toxicological risk assessment usually comes right before this verification phase of the toxicological endpoints, which have been defined in the biological safety evaluation plan. So usually a little bit before, so that laboratory testing can be still influenced with certain parameters. And then for sure afterwards, when the laboratory testing has been conducted, usually a toxicological risk assessment is needed today to finish then the biological safety assessment in general.
Okay, so you start with the overall risk assessment of your device, your body contact, the device classification, duration, etc.
Yes.
And then you start to make a plan. How do I want to test this? Which endpoints do I have to address? Which chemical tests do I need? Because I need chemical data. And then it would be a good point to contact you and see which tests do we really need to perform in order to get our toxicological risk assessment right. And then you get chemical data from the lab. And then you assess what's in the device on a toxicological level and say, yeah, these substances are of no concern. And then there's a few substances that are of concern and that need to be addressed. And then you basically make a report and then you compare report to the plan. And there you go. That's the toxicological risk assessment. Is that a good description?
Yes.
Okay, so what steps, walk us through a toxicological risk assessment, how you would perform that. What steps would you take in order to create a good database for doing that?
Well, to establish a good database for toxicological risk assessment, there's many steps which must be taken usually. And this also depends a little bit on the life cycle of the device, so in which phase we are talking about. But usually, like you mentioned already, the intended purpose, indication, contraindication, contact classification, so type and duration of contact, the patient population, this must be defined or should be known at least. And then, of course, for the patient population, you need to include possible risk groups, and those possible risk groups must be also determined. And then the new revised version of the ISO 10993 strongly emphasizes that this is nothing new, but it strongly emphasizes nowadays that the possible impact of reasonable foreseeable misuse must be also included in the assessment. So you should be also clear what is possible misuse of your device, what are possible misuses of device combinations, and so on, which you can foresee at least reasonable. So this must be also given, because that must also be added to the risk assessment. And this can also affect your patient population. If you know, for example, and that's a very simple example, that your device was actually assigned for grown-ups, 60 kilograms, and now you find out that this is also put into juvenile people, we might have to reassess the toxicological risk assessment due to different body weight considerations than in those cases. Or if you consider patient populations with a chronic disease already and things like this, they might have other requirements or give other input into the toxicological risk assessment, like healthy 35-year-old people. Then the risk management framework is defined. This is also involving then a strong link to the ISO 14971 processes within your company. And then, of course, the risk management plan needs to be updated. And then the biological safety assessment plan must be created or updated. And there is already then all relevant endpoints in it, the test strategy, the acceptance criteria, the links to the general safety and performance requirements. Then we usually start collecting further data from you. So a list of materials and compounds used, for example. We talk about patient contact scenarios. What else? Depending on the risk of the contact scenarios, we might have to include users and third parties. It's rare, but it can happen, especially for combination products, for example, or for drug-releasing compounds. And then, and then we also talk about materials, formulations, additives, coatings, colors and pigments, adhesives, printing inks, whatsoever is put onto your device or touching your device, that is all also influencing it. And then we usually also ask for a short process map to understand that we cover all process-related contacts, such as cleaning agents, lubricants, coolants, release agents, packaging materials, sterilization methods and parameters. Shelf life is also a topic which must be addressed then in the toxicological risk assessment, because it might give us different endpoints where we have to draw samples from. And then supplier data. If you have a lot of steps outsourced, then we must also talk about those. And then, of course, the geometry of the device, exposure scenarios, product families, worst cases within the product families, which must be addressed. And then the analysis is planned. This is usually determining the analytical evaluation threshold, extraction media, extraction conditions. We describe those in detail because the notified bodies, I stick to the European definitions. The notified bodies are looking for those as well, to define that we have set those correctly to really cover a worst case scenario. Sampling times, simulated use then scenarios. We talk about the measurement and the analytical methods and many more. And then we also conduct the literature and inventory, data search. Yeah, and then the necessary documentation must be prepared in this case. And what I want to mention here, usually we talk about the 10993-1, so the famous biocompatibility risk assessment standard, but there's also different standards then which might be considered in addition. So for dental manufacturers, nanomaterials, respiratory equipment, just to name a few, they might have also different biocompatibility requirements and also toxicological endpoints, which must be considered in those cases. So it might be that if we are not talking only about a stainless steel screw, which is an orthopedic device or something like this, that other standards must be considered as well in the whole evaluation.
Yeah, so that's a lot to unpack that you just mentioned there. So let's make a mark at the adult device being used in juvenile, because I have a remark on that regarding MDR and not availability of certain devices. So they're using the adult devices for the children, because pediatric devices are pulled from the market because they make no sense under MDR. It's kind of paradoxical. They want to make the device safer by having higher requirements on the regulatory side, and then they get pulled from the market. And what people or physicians then do is like they use adult devices for children because it's the only thing available to help them. So that's a little a paradox. Not to get too political. We got that once. And then, so you described which data you need. So you need to know a lot about what your device should do, intended purpose, contact duration, and everything that comes into contact with the patient. So everything that goes into manufacturing it, anything that goes into how it's being used and on which patients it's being used. And you need to summarize that in a whole data set that you're documenting, first of all, and then you're deriving a task plan from it, where you use certain extraction methods that we've also talked about in the podcast, in the chemical analysis. And then you try to find the molecules that can come out under the condition that it's used under. And then we look at the molecules that come out. And then we say, yeah, is this toxic or is this not under the conditions that we just mentioned? Is that a good summary?
Yeah, that's a good summary. But usually we do not derive that a substance is non-toxic, because there is hardly something like a non-toxic device or non-toxic substance. That's usually not a toxicology is usually not a black and white picture which you get.
The dose makes the poison. Who was that? Paracelsus or something?
Yeah, the most Paracelsus said that, yes.
Yeah, okay. So let's dive into how you would derive if something is like passes the toxicological risk assessment or not. So we have our device, we analyzed it, we know which substances can be extracted under the conditions that we are using it under and with the patient population in mind. And then we have chemical data. How do we derive if something is, you said it's not black and white. How do we make this analysis, and how do we derive if something is critical, or if something needs to be investigated more, or if something is okay, you can probably put that in a body and be fine?
So first of all, one of the main points to look into first is the contact duration, because the contact duration pretty much defines which toxicological limits we can apply for a certain device, and also for the extractables and leachables coming out of that device. And then we can check if we can apply the toxicological screening limit. We check what is the maximum and total amount of identified substances. We look at the exposure dose and a couple of other factors. And other factors would be, for example, whether exposures accumulate or whether we have added effects which occur on the same toxicological endpoints, because some substances can actually influence each other, and adding up to the toxicological or to the toxicity by having both of those in it. Individually, they would be less toxic, but together they are more toxic. So that is what we take a look into it. And then possible uncertainties and the data quality are also assessed. And then we usually define based on that so-called uncertainty factors, which come into our consideration. So we make the worst case very likely even more worst case. So through those uncertainty factors, to err on the safe side. Then the risk is characterized, and that's usually a mathematical procedure. And from that, at the end, that is the only thing that really our clients are worried about. This is then the so-called margin of safety, because when we derive at the toxicological risk assessment with a margin of safety above 1, then everybody's happy because no further measures need to be applied if done correctly. And then everybody's happy. And if we have a margin of safety below 1, that doesn't mean that the device or the risk assessment is necessarily not valid, but this means that we have to look a little bit deeper and probably refine our toxicological risk assessment in this case. So it in a worst case scenario, we would probably also have to reconduct retesting in those cases if we really talk about very low margins of safety, which are very close to 0. And and then of course everything we do in this scenario must be documented, so it cannot be done undocumented.
Okay, good. So you go through the toxicological risk assessment, take into account everything you know about the device, and then you calculate the margin of safety, and like above 1 is good, and below 1 you need to take further refinements, further measures. Could you explain for someone who's never like dealt with chemical analysis and toxicological risk assessment what the margin of safety describes as a mathematical construct?
In the podcast, okay. Without having a PowerPoint presentation and showing the formula at this point of time, the margin of safety is actually a construct for those who have never needed to deal with it. It just gives you like an orientation of how safe your device is in this case. Yeah, it is not a toxicological value in itself. Yeah, it does not tell you you can take a certain amount of milligrams or grams of a substance and put that into your body and you will survive it. It just tells you how safe we think we are at this point of time. So that is the best thing I can do in a podcast.
I think that's a perfect explanation without getting too much into deep statistics. So let's get over to my favorite topic, and that is sustainable plastics in medical technology. So what I'm investigating since, I would say, many years is if recycled materials can be applied in medical technology. And as you said before, it's always about the risk. And the example I always use is like medical devices between crutches and implants. And you'll never use a recycled material or a mechanical recycled material for an implant. That's probably unthinkable because it's a very risky device. And also the upside of using something sustainable for something that uses a lot less material is probably not that great. So risk to reward ratio, very bad. And then you have something like crutches. That doesn't have body contact because like the patties for your hands are made from a different materials. It's just a mechanical part. So you need no biological safety whatsoever because no patient contact. And there's just no risk involved except mechanical risk. So it can break. And I think there something in between there's the boundary where you can actually use a recycled plastic. And it also depends on the quality of the plastic because like plastics from different sources and recycled plastic from different sources have different qualities. So a solvent-based recycling, which is hyper-purified with the solvent, probably is much more safe and has much less leachables than something that you would use from a, let's say, household trash or something like that. So there's also this uncertainty. But if someone would want to use a high quality mechanical recycled material, let's say PETG blisters that are mechanically recycled from operation rooms, just as an example, what would you do in terms of batch to batch testing and showing in a toxicological risk assessment that this can be done and this is safe? What steps would you take? If you're working in medtech and are struggling to make your devices more sustainable, then there's a link in the description for you.
Okay. So first of all, I want to mention that the usage of recycled plastics is possible. The MDR and the IVDR do not tell you not to use recycled materials as long as you can show that the material is safe and the device afterwards is effective, you can use recycled materials. Now, for the recycling materials or the recyclates, the problem is how they are recycled. Like you mentioned already, mechanically or chemically recycled, there is a big difference. And there's a big difference in the process and in the documentation and in the proof which you must deliver. So we usually talk then about the traceability of the material, how good is that and how good can that be managed, so that there is certain limitations. With regards to your specific problem, the PET whatever packaging materials or polymer materials which are coming out of a hospital setup where we can assume that we have a high degree of pure medical grade polymers collected in those cases. That's possible. Would be depending then on how you recycle it afterwards. And I'm totally going with you. If it was for housings, packaging materials, very likely any medical device that is not contacting breached surfaces of the skin, there you can use it. I don't see any reason why this should not be possible. If your risk assessment, of course, shows that it is safe. So you still need to go through a risk assessment and define those scenarios, and then you can use recycled materials. What I also want to mention is there is currently ISO TC 194 working group, which is looking into the recyclates and into this recyclability of polymers in medtech, because the European Union wants to increase the recycling rate also in the healthcare industry, and therefore there's a working group even, and I think there is going to come out guidance in the near future on how to use recyclable materials, polymer materials in the medtech.
Yeah. Okay. So you need to hook me up with them. I need to talk to them like yesterday, because that's like the topic that drives me. I think medical polymers are often burned, and we have to say like burning polymers is not the worst thing you can do for medical polymers, because I always have the risk of contamination and infection and so on. However, I think a lot of polymers are thrown away or burned, but they are good to use. Like for a single-use PET bottle, you would recycle them 10 times. And for a PET blister that has similar chemical properties, you use it once. So you could probably do something very similar to the PET bottles with the blisters. And that's what my thinking is. So after podcast, I need to get connected to them. But let's move on.
What I want to mention in this regard, when we talk about the recyclable materials, if you can ensure that your recycled materials is coming from chemical recycling processes, then of course you can almost regard it as new. And then you just need to ensure that you really have traceability and control over your supply chain in this case, and proper documentation. But current thinking is in the toxicological community that chemical recycled material is as good as newly made polymer material. Yeah. So that's just
because these polymers are very usually also done in the same plants. They're usually made with mass balance principles, so they're putting fossil-based feedstock along with chemical recycled feedstock. So it is the same material with the same properties. What I also think that has a very high potential, and I'm trying to get someone to speak on my podcast about this for like a very long time, I think 2 years or something. I think I'm going to manage is solvent-based recycling. Because solvent-based recycling is like you purify something with a solvent. It's basically you pre-sort it from mechanical recycling, and then you dissolve it. You take everything out that is not the polymer. You purify the polymer, and then you have a super pure polymer chain that has basically next to no leachables. You always have like the chains that are the molar mass distribution that is not as it is with a virgin. So it's not perfect virgin like, but it's a very high quality. And I think that has also a lot of potential.
I think so too. I mean, for solvent based recycling, you just need to take care that you don't have any impurities in it, or that you can control the amount of impurities. But also there, if you have traceability and quality control behind it, why should that not be possible? But also in those cases, you really need to check carefully where your recycled material is coming from, because, for example, polymers which are coming from the electronic waste streams and housing materials, they can be contaminated sometimes. So, but yeah, recycling is a big topic, and I think also the medical device industry should look a lot more into it. And also, I think that in the medtech industry, we could do design-wise also a lot more things to the packaging materials to reduce, the waste, especially in the packaging area. That's possible.
100%, I can completely agree with that. So now we walked through the toxicological risk assessment, talked a little bit about what you would do in special cases such as recycled polymers, and now that all sounds, to be fair, very complicated. So you probably, and that this is also what the standards tell you and what the regulations tell you too, it needs to be a competent person in apprentices that does this. What qualification does one have to have in order to do something like that? Because I don't think that like everybody can do this and ensure that the device is safe and effective.
So regarding the competency of a toxicologist, the MDR and the IVDR do not specify a particular professional title which you must have to create toxicological risk assessments. It remains the responsibility of the manufacturer, and he can assign the toxicological risk assessment internally or externally to service providers like JLSC, or to any qualified person which he has available internally. The ISO 10993-17 goes a little bit further and states that a toxicological risk assessment may only be carried out by experienced, knowledgeable experts who are familiar with toxicology, medical devices, and the applications, the materials, processes, and also exposure assessments. So they define it a little bit more, what kind of knowledge you must have. And that's mainly where we are coming from. And if you think about what is necessary to become a toxicologist at Jaksch Life Science Consulting, then usually you need to have a finished degree in a MINT subject, so in a mathematical engineering, science related, or science and technology related university degree. We are especially looking for chemists in this area, microbiologists, but also pharmacists, medical students in this area. And then usually you need to take, if you did not finish a toxicological degree at the university during your study times, you need to agree to become one and take an additional step and do another very likely master-related science degree in toxicology, which is specialized in toxicology. And that's usually the way in for JLSC and for how we train, or what the requirements of toxicologists are when we hire them. The reason for that is why we are pushing very hard for this educational pathway is because the competency of the toxicologist is always reviewed, and by having those right certificates behind it, the toxicological risk assessment is usually not questioned then in this case. While when you put just a normal engineer onto your toxicological risk assessment, you might receive further questions and inquiries regarding the competency from your notified body.
Okay, so I was about to ask you about the shortage of specialists, but you just answered the question right away. So what you do is like you hire chemists, pharmacists, biologists, and you take them through another master's degree alongside with work. I assume, yes, and then you get your toxicologists that you have then trained yourself, basically. So you basically take graduates and put them through a trainee program, basically, that makes them such that they're a finished degree toxicologists that you can then use as distinguished experts, basically.
Yeah, we have a program for that in-house at JLSC. So to enable the possible candidates to do this also part-time during work, because they need to. There's only a few universities in Europe where you can finish that degree, and this usually means to be on site. So there's a burden on it. It's a very stressful time. I can tell possible interested candidates. It's not something you do easily, and you need to spend quite a lot of effort into it. But yeah, that's one way. And the other way is, of course, we also hire specialists who have already a title. But yeah, there's also not as many as you would like to have. So you really have to build and create a strong network, and you look out for those kind of specialists all the time, also for their availability. And that's it. There's not so many toxicologists because the burden is quite high. You need to have another university degree. And before you can become a certified toxicologist, for example, you also need to have 5 years of work experience. So it's quite a long time of training and education, which you need to put into to become a toxicologist by profession. But yeah,
Okay.
It's not impossible.
Yeah, as your example shows. So let's talk a bit about you and your company. Perhaps it's a good time to introduce what Jaksch Life Science Consulting offers as services, and just walk us through your services and what a typical project looks like for you. Do you enjoy the podcast? Then subscribe and rate it on your platform, and join the discussion on LinkedIn. Your feedback and comments help us improve the podcast and bring relevant topics to you. Let's form a better world with plastics.
Okay. So, Jaksch Life Science, when we talk about MedTech, we have our ISO 13485 certificate for that, which makes onboarding for our clients quite easy in this case. So that's one part of the service packaging you get. And then usually clients approach us for various projects. For us, it's only interesting if it has a biocompatibility, biological safety, or toxicological background, but then we answer questions in material science, cleaning, packaging, sterilization, reprocessing, in microbiology, and of course, biocompatibility and toxicology to ensure that our clients can have market access or continue with market access for the devices after the recertification procedures. And that's what we do. That's where we are very good for. We can not only do this for Europe, but we do this or we did this in the past for more than 80 countries already. So we know exactly what to do. And usually if we onboard a client, one of our first questions is where you want to market your device and place it onto the market, because there's different requirements, different standards, different legislation which might have to be looked into it. And regarding what does a typical project look like, I would say there is not such a typical project because it depends on a lot of things, life cycle of the product, where you are standing, the size of your company, the maturity of your quality management system, the maturity of your data set, Do you have devices already on the market, similar ones? Is it a new device for you or nothing? Is it a new technology for you and things like this? So there's not really a one-fits-all solution, but the onboarding process starts usually with the NDA. Then we try to arrange for a meeting where we can go into the detail to understand the problem and to understand the workload which might be included in your project. Then of course a quotation is being placed, and then usually when you return the quotation signed to us, we start working on your project within 3 working days. And and there we start collecting the data which is already available to you. If it's a new device and no data is available, we try to understand what materials you would like to use. We collect material data, safety data sheets, technical data sheets. We collect actually everything, review it, do the literature searches in those cases, and just start pushing rocks out of the way and ticking boxes so we can put together a proper biological safety report and toxicological risk assessment afterwards. And duration wise, how long does it take us? That is also not so much connected to the classification of your device, but actually regarding the properties of your device. So typical projects like this run for between 4 and 2 years, depending on the amount of tests which have to be conducted. Example for a very long-running project is we had lately a client coming with an absorbable device, new material, or at least a new material for the application. So there was not such a thing like a predicate to be used, which is forcing us to go into an animal study setup, which is running then for 18 to 24 months. So this is how the timelines are coming along. Yeah, and this is all what we put into consideration. Also, what we put into consideration, this is what I mentioned already, we take a look into which markets you want to place your device on, because the gap is actually widening at this point of time between the US requirements and the European requirements due to changes in the -1 and a couple of the other 10993 standards. So there's a huge gap and acceptance gap also in terms of data. And this is all considered by us. So if you tell us, for example, you go to the US, we will prefer a more in vivo based test strategy. For example, if you tell us you want to stick to Europe and maybe some Asian countries, then we might stick more to an in vitro based test strategy in those cases. So this is all collected and all formed into then the services you get from us.
Okay, cool. Thank you for the insights. So you founded Jaksch Life Science Consulting 10 years ago, and you've grown in the last 10 years. How did you experience that journey?
Well, JLSC is getting 10 years this year in December. So I need to say I had a very good time in those 10 years. It was very challenging, not just serving our clients, but also growing JLSC. It's the speed is very high. It's always very intense here. And actually, I just want to say thank you to the clients who trust us and who put long-lasting relationships together with us. And I would not like to miss one of them, and I would not like to miss a person on the team or former team members which we had. It is it's a very intense time, and I'm really looking forward to the next 10 years, because I think it's going to be challenging also in the next 10 years. Yeah, there is new sustainable materials coming up, data-driven decisions also in toxicology, AI tools which might have to be included. Yeah, drawing closer also the collaborations with our clients. There's quite a lot of exchange with them. We know them. It's like, it's the journey will be continue to be great. So that is really what I'm looking forward to.
Yeah, that's that was nearly perfect last words, but I just want one last question, because that is what I ask everybody. Is like, what advice would you like to give to our listeners in regards to toxicological risk assessment?
Actually, I don't have advice. I just can tell you you cannot do without one. So you need to do it, and you need to find a way to deal with the requirements, as much as you might not like the outcome and the results. But yeah, you need to do it. There's no way in delaying that unpleasant topic, and get done with it early. Include the toxicologists early, because that will save you time and money at a later point of time.
Yeah, is that goes in line with a lot of advice that we're hearing here. It's like communication is key. Involve the people, involve the key people, and then it's going to get good. But you need to involve them in an early stage. Involve the experts. Do you have any other last words you'd like to give?
No, I just want to say thank you one more time to you, Lucas, the Gradical team, and of course to all you for listening. Yeah, and if you're interested in risk-based biocompatibility, biological safety assessments, or toxicology, reach out to me or reach out to Lucas. And yeah, thank you very much, and I see you next time.
Yeah, that's this was Arne Jaksch from Jaksch Life Science Consulting, and until the next time. If you're working in medtech and are struggling to make your devices more sustainable, then there's a link in the description for you.